Okay, let's unpack this. When we talk about cancer treatment, immunotherapies, especially these anti-PD-1 therapies, have just offered so much hope, haven't they?
Absolutely. Transformative for many. Gave people real possibilities where maybe there weren't many before.
But, and it's a big but, they don't work for everyone. That's the real challenge we're still facing.
Exactly.
It's not if they work, it's the mystery of why. Why are they, a magic bullet for some patients, but seem to completely miss the mark for others?
Right. And that critical question is really what today's deep dive is all about. We're looking at a really important paper from 2018 in Nature, Chen and colleagues.
Right.
And what they did was uncover this, surprising way tumor cells can hide from the immune system, a mechanism we didn't really recognize before. And even more crucially, it points towards a new way to maybe predict who's going to respond to anti-PD-1 therapy, specifically for metastatic melanoma in this study.
So understanding a hidden enemy, but also finding a potential predictor.
Precisely. That's the goal today, to really get into this hidden mechanism and see how it might be a game changer for predicting treatment success.
Okay, so maybe let's start with the basics. Just briefly, how do these tumor cells normally try to outsmart our immune system?
Well, one of the main ways is through this protein called PD-L1.
Right.
The tumor cells display it on their surface. It's essentially like they're holding up a sign saying, "Don't eat me."
A stop sign for the immune system.
Exactly. And that sign interacts with a receptor on our T cells, our main immune killer cells, called PD-1.
Okay.
When PD-L1 binds to PD-1, it's like a handshake that basically tells the T cells stand down. It inactivates them.
So the killer cell just stops killing.
Pretty much. And that's where the current therapies, the anti-PD-1 antibodies like pembrolizumab, come in.
Right, they block that handshake?
If they block the handshake, they get between PD-L1 and PD-1, so the T cell doesn't get the stand-down signal. The idea is to, reinvigorate the T cells so they can attack the tumor.
Makes perfect sense conceptually.
It does. But here's the problem this research really zeroes in on. Even with that understanding, the actual response rates in the patient group they studied, metastatic melanoma patients, were surprisingly low.
How low are we talking?
In this paper, they mention rates around 4 to 5%. Now, that's specific to this context. These drugs work better elsewhere. But for this group...
4 to 5%?
Yeah. It tells you something significant is missing, right? If we're blocking the main signal, why isn't it working more often? What else is going on?
Exactly. What are we missing? What did Chen's team find?
Well, this is where it gets really interesting. They looked beyond direct cell-to-cell contact.
Okay.
Cells, you see, they don't just talk by touching. They also release these tiny little packages like messenger bubbles wrapped in lipids. They're called extracellular vesicles, and a key type is exosomes.
Exosomes. Heard of them. Tiny messengers.
Right. And what this study discovered was PD-L1. It isn't just stuck on the tumor cell surface.
Wait, it's on the exosomes too?
Exactly. Imagine the tumor cell not just holding up its "don't eat me" sign, but also sending out countless miniature versions of that sign floating through the body inside these exosomes.
Wow, so it's like sending out decoys. Spreading the signal far and wide.
That's a great way to put it. And they proved this rigorously. They took these exosomes from melanoma cell lines, both primary and metastatic, verified them with high-tech methods, electron microscopy, nanoparticle tracking, all that. And the key finding, exosomes from the metastatic cells had way more PD-L1 than those from primary cells.
Okay, so the more aggressive cancer is better at sending out these decoy signals.
It seems so. And they also found PD-L1 on other vesicles, microvesicles, but less of it. Importantly, they confirmed the PD-L1 on these exosomes has the same structure, the same part facing outwards, as the PD-L1 on the cell.
Meaning it could definitely bind to the T cell's PD-1 receptor.
Correct. It's fully functional. And here's another twist. You know IFN gamma, interferon gamma.
Yeah, the cytokine released by active T cells, right? Part of the immune response.
Exactly. Well, IFN gamma actually caused the tumor cells to load more PD-L1 onto these exosomes.
Get out. So the immune system tries to fight back, releasing IFN gamma.
And the tumor responds by sending out even more of these immunosuppressive exosome signals. It's an adaptive response by the tumor.
That is incredibly sneaky. It's like the tumor is using the immune system's own weapons against it.
Precisely. And they even dug into how the cell does this. They identified specific cellular machinery, proteins like HRS and RAB27A, involved in packing the PD-L1 into these exosomes.
So it's definitely not just accidental leakage. It's an active, directed process.
Absolutely. A deliberate strategy, it seems.
Okay, so the tumor's flooding the system with these PD-L1 exosomes.
Yeah.
What does that actually do to the immune response? How does it help the tumor survive?
Well, it actively suppresses those crucial CD8 T cells. And importantly, it does this systemically, far beyond just the immediate area around the tumor.
Systemically, how do they show that?
They observed the exosomes physically interacting with CD8 T cells, especially the activated ones, the very ones you need. And when T cells were exposed to these PD-L1 exosomes from melanoma cells, their key functions just plummeted.
Like what?
Their ability to multiply, to proliferate, their production of essential anti-tumor signals, those cytokines like IFN-gamma, IL-2, TNF, and critically, their ability to actually kill tumor cells' cytotoxicity.
All knocked down by these little bubbles.
Knocked down significantly. And the clincher. If they pre-treated the exosomes with an antibody that blocked the PD-L1 on them, those suppressive effects basically vanished, strongly confirming it was the exosomal PD-L1 doing the damage.
Wow, okay, that's convincing in the lab. Did they see this effect in, living models?
They did. In mouse models with human melanoma, the levels of this circulating exosomal PD-L1 directly correlated with how big the tumors grew.
More exosomes, bigger tumors.
Seems that way. And when they actually injected these exosomes into mice, the tumors grew faster. Not only that, but the number of active fighting CD8 T cells dropped significantly.
Oh, yeah. Just near the tumor.
No, that's the key. The T cell numbers dropped not just at the tumor site, but also systemically in the spleen, in the lymph nodes.
So these exosomes are traveling and shutting down the immune response throughout the body.
That's what the evidence points to. It's a systemic suppression. And they also found similar PD-L1 exosomes, also boosted by IFN-gamma, in lung and breast cancer cell lines too.
Suggesting this isn't just a melanoma thing. It could be a common cancer tactic.
Very likely a common and quite dangerous evasion strategy.
Okay, well, it's a bit terrifying, understanding how cleverly the tumor can fight back on a systemic level. But let's pivot to the hope here. Can we actually use this knowledge? Can measuring these exosomes help patients?
That's the really exciting part. The study found that measuring circulating exosomal PD-L1 could be a powerful biomarker, especially for predicting response to pembrolizumab in these melanoma patients.
How so? What did they find?
Two key things, really. First, looking before treatment started, patients who ended up not responding to the therapy, they had significantly higher levels of this exosomal PD-L1 in their blood before they even got the first dose, compared to the patients who did respond.
Higher levels beforehand meant a worse outcome.
Exactly. It suggests that maybe if the system is already flooded with these suppressive signals, the T cells might be too, let's say, exhausted or overwhelmed to be properly reinvigorated by the anti-PD-1 drug.
That makes a lot of sense. Okay, what was the second finding? This sounds like the crucial one.
It really is, potentially for monitoring patients. This is the bit that seems a little counterintuitive at first.
Okay, I'm ready.
In the patients who did respond well to the therapy, their levels of circulating exosomal PD-L1 actually increased significantly within the first 3 to 6 weeks after starting treatment.
Increased. The bad signal went up in the responders. How's that work?
I know, right? But the non-responders didn't show this increase. Their levels stayed pretty flat.
So the increase was the good sign.
Yes. They even identified a specific threshold, a fold change greater than 2.43 at that early time point that strongly predicted who would respond.
Wow.
A higher increase was linked to better response rates, longer progression-free survival, better overall survival. It was a powerful predictor.
Okay, so help me understand the paradox. Why would more exosomal PD-L1 signal a good response to a drug that blocks PD-1?
Think about what's happening. The drug, pembrolizumab, is working in responders. It's blocking the PD-1 pathway, waking up the T cells.
Right.
Those newly activated T cells then release IFN-gamma.
Okay.
And what did we establish IFN-gamma does?
It tells the tumor cells to make more exosomal PD-L1.
Exactly. So the increase in exosomal PD-L1 is likely the tumor's desperate adaptive response to the effective therapy. It's pumping out more suppressive signals because the immune system is hitting it harder.
I see. But because the therapy is also blocking the PD-1 interaction on the T cells.
That extra exosomal PD-L1 becomes essentially futile. It can't effectively suppress the T cells anymore because the drug is in the way. The increase is a sign the battle is truly engaged.
So in non-responders, the lack of increase means the T cells probably weren't activated properly in the first place, or maybe the tumor has some other way to resist.
That's the thinking. Either the initial T cell response wasn't strong enough, or perhaps the tumor cells weren't responding to the IFN gamma signal for some reason.
That's a really elegant explanation. The increase isn't the problem. It's a sign the drug is causing a reaction.
Precisely. And importantly, they found that measuring this circulating exosomal PD-L1 was a much better predictor than looking at other ways PD-L1 is measured, like total circulating levels or levels on other vesicles or just loose in the blood.
Which points towards a practical application, right?
Absolutely. It strongly suggests we could develop a simple blood test. Measuring exosomal PD-L1 levels before and during treatment could give doctors a dynamic, real-time view of whether the therapy is likely working.
Much less invasive than repeated tumor biopsies.
Way less invasive and potentially much more informative, especially since PD-L1 levels can vary a lot within the tumor itself. A blood test gives you a more systemic picture.
So really wrapping this up, you can see how this deep dive into Chen's work just gives us such a clearer picture. We go from understanding this really subtle, kind of sneaky way tumors evade the immune system.
Right, using these exosomes.
All the way to finding a tool, a biomarker in the blood that could potentially predict who benefits from a major cancer therapy and monitor their response early on.
It really connects the dots from basic science to potential clinical impact. It shows how we can get this live feed almost of the fight happening between the tumor and the immune system by looking at these exosomes.
It's a fantastic example of how digging deep into the mechanisms pays off.
Definitely. And it leaves you thinking, doesn't it? This study focused on PD-L1 on exosomes. But what other messages, what other hidden signals are tumors sending out via these vesicles that we haven't even started to decode yet? And if we could uncover those, how might that lead to even smarter, even more precise ways to fight cancer in the future? That's really something to consider.